my postdoctoral mentor. He's a professor at Cedars-Sinai Medical Center and University of California, Los Angeles. And I spent 5 years with him there, and many an hour having coffee and talking really big picture science. So I think, you know, I could go really on and on about his accolades and some really fun facts, actually. So he sat on the thesis committee of Dr. Pagliassotti. Is he here? Oh, no, he had to go to Denver, actually. So he was able to meet with him this morning and reminisce about those old days. He also was Dr. Heather Pidcoke, our new medical officer, and they were neighbors in Los Angeles. So he has a lot of really nice and kind of small world connections to Fort Collins. So it was hopefully good for him to kind of meet all those old friends, and we're going out to dinner tonight with Dr. Pidcoke. So yeah, I just wanted to mention a couple things. Like I said, I could go on and on about accolades and hundreds of publications. Also in 2006, Dr. Bergman was awarded the Banting Medal for Scientific Achievement, and this is the highest honor that can be given by the American Diabetes Association. So really a wonderful scientist, but really has been an exceptional mentor to me. So he's still the person. He was actually here 1 year ago for our lab opening. I don't know if you remember, but he played some musical entertainment at our lab's grand opening. So I think maybe a yearly tradition might be kind of nice to come visit and talk me off the ledge if I get bad grant reviews. That's what I said. In 2 weeks, I'll be calling him crying. So yeah, so I mean, we were together in Los Angeles for 5 years, which included the birth of my first child and you know, really big life events. We were at 2 different institutions together, USC and Cedars. Dr. Bergman would call me at 3 in the morning. Don't worry, not when I was home like, to bug me, but when I was doing experiments and he knew I was up with the dogs doing 24-hour sampling, he would sort of call me to keep me company and say hi and kind of be solidarity for us doing experiments overnight. So I've really turned into a wonderful friend over the years and just an exceptional mentor, really wonderful supporter of women in science, especially being someone who had really young kids when we worked together. So I've always appreciated that, and I'm just really honored that he can be our kickoff speaker. So with that, thank you for being here.
She can give me my mic. Well, it's always something. Anyway, thank you very, very much, Josie. Josie and I, we had a wonderful time in Los Angeles working together. It was louder. Well, that's what that's supposed to be.
I know, sorry.
We had a wonderful time. And I was an actor as a child. Very few people know that, so I learned to speak louder than necessary. I should say a word about Heather. Heather was, we weren't just neighbors, we were right across the street from each other, maybe 30 feet away, and she was the babysitter for our children, and we now have 5 grandchildren, so I'd say that was a very successful babysitting.
Didn't kill them.
Didn't kill them. Anyway, it's wonderful to be here. I've had a wonderful experience today. It's been great to talk to all the students about what they're doing and try and give them a little bit of perspective from a, let's say, senior scientist. I use that as a euphemism. And I always tell my students that nostalgia is the only pleasure which is reserved for old people. And so I told them a little bit about my career. So today, because I know there are a lot of students here, a lot of times when we talk in medical schools and there's just faculty there with grim looks on their face. But today I'm going to try and give you a little bit of understanding about how the work we did has developed over the years. And we go back to the very beginning and then end up with the latest stuff we're doing and try and show you that it's really it's related to each other. Now, one thing I know here is that in order to go forward, you have to press the backward button, and I was able to do that, so I'm still cognitive, as they say. So people are interested in the pathogenesis of type 2 diabetes. We're interested in it, and there are a variety of ways to look at it, and precision medicine, as you know, is trying to decide to design certain kinds of treatments for individual patients. And that's usually done with biomarkers you're all familiar with, as well as the genetic approach, doing GWAS, trying to find the risk of who's got what's the risk of diabetes, what's the risk in individuals. And in certain diseases, of course, that's been very, very useful. But what we do is something different. We use a physiological approach. And all my career, all the time that I've worked in Northwestern and in Los Angeles, we've done physiology, and we tried to use it to understand what the pathogenesis is, what's the series of events which go forward to make diabetes develop. Now, what have we learned from the GWAS study? Well, most of the gene variants, type 2 diabetes, are associated with impaired insulin secretion, not insulin resistance. This was a big surprise, because people thought they'd find genes for insulin resistance, but they didn't. We have 403 genetic variants, so therefore the holy grail of finding an additional, a particular gene for type 2 diabetes didn't happen. In fact, the University of Chicago was the first, Graham Bell had the first gene which he called NIDDM1. He said, this is the gene for diabetes. Everyone got very excited. It turns out not to be true. It wasn't just that gene, it's not even one of these 403 genes. So even if you try and develop a risk factor profile from the genetics, you can only account for about 20% of the risk of type 2 diabetes. And so that made us want to move into other directions. Now, what is the approach that you can use? Well, this is the kind of molecular approach that people are using. We use this onion to represent individual cells. The layers of the onion are taken apart, and we try and figure out what's really going on underneath the onion. And you can see that we use these different approaches: physical molecular biology, fractionation, kinetics, in vitro activity, cloning, GWAS, different omics approaches, and I'm sure that's what 90 or 95% of the people that you meet are going to do. They're looking at individual genes, they're doing knockouts in mice, they're trying to figure out what the pathogenesis of diabetes is, and I must say, even after all these years, that was the primary method that people used to understand the pathogenesis. But I'm a little bit of a iconoclast. I don't think that can be denied. I was trained as an engineer, not as a biologist. I then went into physiology in graduate school, and so all my career, with all of my great fellows, including Josiane, we have actually tried to study the whole system, not the individual cells. So as a kind of a metaphor for that, you can think of this device, which used to be called a camera, and then we do an experiment and we test how that device works. We define hypotheses, we define experimental designs, we use mathematical modeling. You do all these things at once. And so one of the themes of this talk is to talk about how we integrate all those different approaches to understand how blood sugar is regulated, and then what's wrong in metabolic function, what's wrong in diabetes, what's wrong in other kinds of metabolic diseases. So here's the overlying method. You can see that the process of the scientific method involves making conjectures or hypotheses, deriving prediction from them, logical consequences, and carrying out experiments or empirical observations. This used to be called the scientific method. So basically, you make a hypothesis, you design an experiment, you then see what's wrong with the hypothesis, and you continue to do this until you find the answer. In our case, it's a little bit different because we not only have hypotheses, the hypotheses make predictions, we then design experiments, but we also include modeling, as I'll show you, because modeling is another way to test what the value of the hypotheses really is. This is different from what other people do because they don't always include modeling in this process. But I'll give you examples of how that can possibly work. Now, the first thing you need is some data. And this particular figure goes all the way back to when I was at Northwestern University in the early 1970s, when we were the first ones to really inject glucose into animals and measure every minute. That had never been done before. So people thought that when you injected glucose into somebody, it would come down exponentially. It didn't come down exponentially. It had a very funny shape. And in modeling, the funnier the shape, the more you can learn. So basically, these are just the first 6 intravenous glucose tolerance tests ever done by a student in my lab at the time, Y.Z. Eider. And you can see, if you look at the glucose on the top, they're quite variable from one person to another. And what's shown on the bottom are the insulin. Sometimes you get a very big first phase of insulin, there's a second phase, And you can see these three different animals had very different patterns. So what we tried to understand is what can you learn from these patterns? What can you learn about how the blood sugar is regulated? Well, the first thing you have to know is something about blood sugar regulation. So in those days, and ever since, we knew some very simple things about how the sugar was regulated. And if you think about what you're like in the morning before you eat anything, your blood sugar is possibly normal if you're a normal individual, and that's because your liver is making glucose and your tissues are taking it up. So in the morning, you have a balance between the production of glucose and the utilization of glucose. And they're equal because your blood sugar is constant. So we knew that, everybody knows it, and the uptake of glucose in the morning is independent primarily of insulin. The insulin is very low. Most of the glucose uptake is about half of it is by the brain, the other half is by other tissues, and it's insulin independent. I'm going to talk more about insulin independent uptake and production a little bit later. So we have something that, this is a word that I made up and now it's in the literature. By the way, I should tell the young students, you want to make something up that has your name on it. This is very, very important. So this doesn't have my name on it, something else does. But we called it glucose effectiveness. It's still a pretty lousy word, but that's what people use. So glucose effectiveness is the ability of glucose itself to stimulate glucose uptake by the brain and other tissues, and to suppress glucose production by the liver. So the liver is making glucose, but it's making it at a limited rate, and glucose suppresses that, so it's not too much production. If you have too much production by the liver, of course, you can have type 2 diabetes. Now what happens when you eat your meal, you eat breakfast? Well, you all know what happens. When you eat a meal, it stimulates the pancreas to secrete insulin. So the pancreas makes insulin, and then it goes into muscle tissue and other tissues. So the insulin goes up in the blood, it's transported in other tissues, I'll talk more about that, and then it goes into the interstitial compartment. And that's a very important fact that I'll talk about later. And once insulin is in the interstitial compartment, it can stimulate glucose uptake by muscle. So insulin goes from the blood across the capillary into the interstitial compartment. It binds insulin receptors on muscle and causes glucose uptake. It also suppresses fat production by adipose tissue, and it also turns off the liver. So when you eat a meal, the liver is then turned off. First the blood sugar comes out, the blood sugar goes up, then it turns off the liver, increases glucose utilization. That's called a closed loop feedback system. As you all know, we've all learned about that. Well, it turns out engineers study closed loop feedback systems all the time. So for me, this was a very interesting model to study because I could study closed loop regulation of the blood sugar and try and understand how the sugar is really regulated. So what we did was to ask the question, how can we model this? And I'm not going to give you very many equations. Don't get nervous. There are probably some engineers. Are there engineers? Any engineers here? A total of 0. So, but you should bring some engineers in. It's very important to have a few around. But anyway, what we did was we asked, what's the simplest model that can account for the blood sugar regulation I showed you before? Remember the time course of glucose and insulin. And we started with the simplest model shown in the top left, which says that there's a production, that's the P, of glucose by the liver. There's the uptake of glucose, that's what the K is. And we just said we can write an equation for that. Well, it didn't work. So that equation was inadequate. So then we started to look at more complicated equations. Number 2, we included a Michaelis-Menten term. That didn't work. So we started to slowly and systematically complicate these models until we found one that worked. And that was a very good process, which hadn't been done before. Because we asked, what is the simplest model that accounts for glucose-insulin kinetics? What is the simplest model that can do it without error? And it turns out there was one model that worked. Now, what are the characteristics of this model? Well, you can see we're not modeling the secretion of insulin. We're only modeling insulin's effect and glucose's effect. So what you can see in this model, I'll try and explain it to you. Again, I have to do it this way. It's always this way. It's one in the middle. Okay. So here you can see, why did you tell me this is red? Isn't it green?
It's red.
I'm colorblind.
He's colorblind.
8% of all men, it's not so bad. So here you can see the way this model works is the liver makes glucose. So this is the glucose in the blood. Liver makes glucose here, goes in there, and then the glucose goes into the peripheral tissue and takes it up. Now what does insulin do? Well, what insulin does is it stimulates the uptake of glucose, and suppresses the production of glucose. So insulin does both of those things. That's why your blood sugar comes back to normal after you eat a meal, because it activates the peripheral and suppresses the production. But the interesting thing that we learned from this model, which is very, very important, is it isn't just the insulin in blood that does this, it's the insulin in a compartment remote from blood. In other words, there had to be a delay in the insulin action. So this we call the minimal model. And these are the equations. There are no engineers here, so I'll skip it. So here's the minimal model, again shown as little bubbles. Is there anyone here named Bubbles? No. So what happens when the glucose goes into the blood? It goes into the blood, it can turn off the production, stimulate the uptake. That's a bad sign. I don't know how that happened. So the point of all this, and what we learned from this model, is that insulin action in blood is delayed in time. And that is true. Insulin doesn't work immediately. So if you inject insulin, if it goes up during the test, anytime, it takes time for the insulin to work. Now why is that? Well, we hypothesized that the delay in insulin was the time it took to get from the blood into the interstitial fluid. This is to cross the capillary endothelium. We said that, we hypothesized that that's why insulin's work was delayed. It was a hypothesis. Now remember, we started with data, went to a model, and the model led us to a hypothesis. This is the process that I'm trying to describe to you. So we then had to test it. So this is very important. If you know people who model, if you have friends that model, they have to validate their models. There's no point in just publishing a model, saying it's right, without validating. So we had to validate it, and it was a lot of work. How did we do that? Well, we figured we had to measure the interstitial fluid and see how the time course of insulin action is related to insulin in blood or insulin in the interstitial fluid. Experiments. So what kind of system do we use? Well, what we did was we were able to put a catheter in the lymph. This was in this part of the leg, so that we could measure interstitial fluid, because the lymph is interstitial fluid. We could measure the interstitial fluid here, and we could give insulin on the arterial side. It goes to the venous side. Meanwhile, we could measure interstitial insulin. We could look at the temporal relationship between insulin in blood, insulin in interstitial fluid, and glucose uptake by isolated muscle. And we published a lot of papers on this, and we're still working in this area. So, that's right. This thing's back. So what does this tell us? Well, we did this study where we actually injected insulin and measured insulin in the blood, measured insulin in interstitial fluid. So here's the blood insulin with an insulin injection, which we measured. And here is the insulin in the interstitial fluid, which you can barely see on this picture. If you look over there, you can see it. And then we measured the rate of glucose uptake. And we found that the insulin in the interstitial fluid had the same pattern as the glucose uptake. Remember, the blood insulin does not have the same pattern. It's very different. So this proved that the delay in insulin action in the model was due to transport of insulin into the interstitial fluid. And in later studies, we showed that in obesity and diabetes, this transport rate is changed. So this is an example of how we used an experimental test. We had an original observation, we had a model, it made a prediction, and then we then confirmed the prediction with an experimental test. So this is then what happens. Insulin goes into the capillaries, and then it crosses the endothelial barrier. This takes time. You end up with insulin in the interstitial fluid. It then binds to the receptors on muscle, and then causes glucose uptake. That's turned out to be true and very important. So the putative daily insulin action is the time to cross the endothelial barrier. So then the question was, are there clinical applications of this minimal model? Up until about 1980, we had just really were doing so-called basic science. We didn't have any clinical applications. But we began to realize that that was important because people became very interested in those days in insulin resistance. Insulin resistance is a concept that goes back to the 1930s, but it was only in the 70s that people became really interested in insulin resistance and its role in type 2 diabetes. I'm sure everyone in this room knows that insulin resistance is an important feature of those people who have type 2 diabetes or have impaired glucose tolerance. So we then invented a clinical test. So this test is called the Frequently Sampled Intravenous Glucose Tolerance Test, or FSIGT. I'm sorry for the long name, but the point is that we simply inject glucose and then we inject insulin 20 minutes later. I won't go through why we chose that protocol, but it's very easy to understand. So time 0, glucose is injected, shown here, and then 20 minutes later, insulin is injected. And when we inject the glucose, you can see the blood sugar goes up very, very high. Those are the blue dots, and then it comes down, and that's because it's mixing in the blood. And then we inject insulin, the insulin level goes up, and it accelerates the declining glucose. First it comes down at a slow rate, and then after you inject insulin, it comes down at a much faster rate, and then after about 4 hours, it's all renormalized. So then we have a time course of glucose and insulin, and they don't look the same. The glucose goes up very fast, it comes down, then there's an endogenous insulin response, an exogenous insulin response, biphasic, accelerating the rate of fall of glucose, and that goes on for 4 hours. Now what's the advantage of doing this kind of test? Well, we have the minimal model. Remember the minimal model? And we have it implemented on a computer. So we can tell the computer what the glucose is and what the insulin is, and the computer will tell us what insulin resistance is. That's the advantage of the model. So first we use the model to a test of the delay question. Then we could apply the model for clinical purposes. We could do this test in humans, which we did, take the data, give it to the model, and it'll calculate things that the model can find out in that process. So we put the data into the computer. It doesn't have to be done at the same time as the test. And then the computer tells us two things. The response of the beta cells, in other words, the insulin secretory response, and the insulin sensitivity. Those are the two things we were interested in. Why? Because people were arguing about which was important, and this Mike probably decided to remember these discussions, which was important in the pathogenesis of diabetes. Is it the insulin resistance, or is it the beta cell response? You go to meetings, the ADA meetings, and people would scream at each other about this idea. Dan Port would be screaming at Jerry Regan about which one it was. The Seattle people were sure that it was insulin secretion, whereas the Joslin people were sure that it was insulin resistance. It was very accurate. So by modeling this, we were able to do what I just told you. We were able to address this question. Which is responsible for type 2 diabetes or metabolic syndrome? Or is it beta cell function or is it insulin resistance? Remember, we got these two different groups. But by modeling it, we're able to look at the two functions at the same time. And I think we were the first ones to really do that. So we ended up with this hypothesis. Now remember, we had the delay hypothesis. This is another hypothesis, and this one just came out of the air. I said, I think that this relationship is going to be hyperbolic. Why? What does that mean? Well, that means that if you have a person that's very insulin sensitive, for example, all the exercise nuts in this room, you're very insulin sensitive, and you don't have to secrete much insulin, as you know. If you then, over the course of, let's say, the holidays, you eat too much, you become more resistant, why don't you get diabetes? Because you're normal. You don't get diabetes because you secrete more insulin. So therefore, what happens is your insulin sensitivity goes down, you become resistant, and then the insulin secretion goes up. So you move from this point on the curve right there to the other point. Now you also become resistant when you get pregnant. When a lady is pregnant, she becomes resistant. When people gain weight, they become resistant. During puberty, we all became resistant. So there are a lot of normal physiological functions that cause insulin resistance. What happens then is your body, which is very clever, secretes more insulin to compensate for it. It also does something else I'll talk about later, but I don't want to use it here. So I hypothesized, and I can't tell you why, that the product secretion sensitivity is a constant. You remember from high school that x times y is a constant, is a hyperbola. It's this curve right here. So I hypothesized at the time, along with my colleagues, that this will always be true. If you become more resistant, you secrete more insulin, you stay on the same curve. And this is a curve, a hyperbola, which is described by this very simple equation. Now, that hyperbola which is defined by one number, I call it the disposition index. And if you look at literature now, there are about 1,000 papers citing the disposition index. So it's an accepted fact now in diabetes that the relationship between secretion and sensitivity is one number. Now, if you're a person who's has really great beta cells, you'll be on a high curve up here. But if you're a person who's at risk for type 2 diabetes, you'll be on a low curve here. What does that mean? That means for a given change in insulin sensitivity, you'll compensate much less and you won't go as high as if you have great beta cells. You'll go there. And we've showed in many, many different cases, and other people have shown that the disposition index is a great describer of your metabolic function. Now, what does it tell you? It tells you how well your beta cells compensate for insulin resistance. So we think this normalized the argument about which one is it. It's both insulin secretion and insulin sensitivity, insulin resistance and reduced beta cell function. And you can describe it by a higher or lower disposition index, that is which curve. Now, once in a while you wake up and you go into the office and you're in a bad mood, and you turn on your computer and something comes up, and that's what happened to me. This paper, where this group had looked at this hyperbolic relationship in a whole variety, there was a meta-analysis, a whole variety of different groups who were African American, Asian, East Asian, and they all more or less lie on this hyperbolic curve. So they showed, and of course, the dots represent which group it is, they represent how many people were in the study, but basically it confirmed that the hyperbolic relationship is almost true. Now, as you can see, at the very low level of insulin resistance, it doesn't quite fit the curve, and I guess I could spend the rest of my career trying to figure out why that is, but 99% it fits the curve. So again, here was a hypothesis based on the model, which then could be tested experimentally, not just by us, but by many other people. And it confirmed that the hypothesis was fundamentally accurate. And if you look at the prediction of diabetes and use the disposition index, it's a much better predictor of who's going to get diabetes and other things. So everybody in this room has a disposition index. If we do that frequently sampled test on you I described before, we'd figure out what your disposition index was. If it's low, you have a higher probability of diabetes than if it's a high. So it's a predictor of who's going to get type 2 diabetes. And it's a stronger predictor than a lot of other clinical measurements. And that's shown by a lot of studies. This is one on Christian Beyer and Cliff Bogardus on the PIMAs, which are in Arizona. And what they showed is they measured insulin sensitivity and they measured beta cell response. And they looked at two different groups of PIMAs who had slightly different disposition indexes. So the ones that are on these upper curves on their NGT, they were normal. They had a pretty good disposition index. There was another group that had a lower disposition index, and they're down here. I can't see this thing. And they're down here. So you can see that they had very different outcomes. The ones that had a higher disposition index after 5 years didn't get diabetes. The ones that had a lower disposition index, first they went through IGT, and then they got diabetes. So this shows the disposition index is a predictor of type 2 diabetes. And now it's been shown that you inherit your disposition index from your parents. It has a strong heritability. So from your parents, for better or worse, don't blame me, you've got a good disposition index and not such a good one. If you have a lower one, you have a higher risk of diabetes than higher. So that again is another example of a hypothesis which has been tested and shown to be more or less accurate. Nothing is perfectly validated in biology, but it fits pretty well. Well, then the question arises, why this hyperbola? Why does this happen? What is the underlying mechanism? And to this day, we don't actually know the answer. You may go to your friends who are diabetologists, Dr. Hooper, for example, and you may say, Dr. Hooper, or also Dr. Hooper, you may say, why does the beta cell know how to compensate for insulin resistance? Your beta cell does that every single day. And if you do an exercise regimen, it does a better job. Or if the lady becomes pregnant, it does a worse job. But in every case, we don't know why the beta cell knows what to do. After every meal, the beta cell knows how much insulin is secreted. So we looked into this question, and we couldn't totally find the answer. And we did these cross-sectional experiments, and this is in dogs now. This is hyperbole not only applies in humans, it also applies in dogs. So here's dogs, fed on a high-fat diet, so they become insulin resistant, and they secrete more insulin. That's what it's supposed to do. Dogs can't climb, but they climb right up the curve. So the question is, why is that? So we looked at all the metabolic changes that happen. I need some water. We looked at all the metabolic changes, that is if you want me to go all the way to the end. All the metabolic changes that occur when we fed high fat to these dogs. Well, that turned out to be very, very interesting. Because you can see that after feeding fat to the dogs, their body weight went up. That wasn't surprising. Their fat deposits both in the visceral depot and the subcutaneous depot, which is also not surprising. Visceral depot being the belly, subcutaneous everywhere else, and the dog mostly on the back. And then we measured the glucose. And to our great surprise, during this period, the dogs are insulin resistant. They go up on that curve, their glucose doesn't change. This was a shock. And believe me, the community did not believe it. And they said, well, you just didn't measure it carefully enough. You didn't measure it enough times. So we finally had to do a study, 80 animals, and we measured the glucose. We showed, with a method that was good to 0.1%, the glucose doesn't change. If the glucose doesn't change, how does the beta cell know what to do? We think it would look at the glucose, but that isn't what happens. So with this fat diet, you can see that the fasting insulin goes up, that the insulin sensitivity goes down, so they become more resistant, they have more insulin. The insulin clearance goes down, and this is something we're talking about a lot more, which means that the disappearance of insulin from the blood is reduced. So they're hyperinsulinic because they secrete more, and they have a higher insulin because they clear less. And finally, the first phase insulin change. So the question was, what's really going on? If the glucose doesn't change, why is it that these animals, normal animals, fed fat, do what to do? Well, we measured a million things. These were overnight studies done similar to what Josie did in the lab, but this is before she was there. Thank you. During these studies, all the people who did the studies they looked at Sex in the City. They got videos of Sex in the City, and they watched them overnight, and they sampled every hour. And we measured many different hormones to see what the signal could be. And none of the hormones we guessed could be the signal actually changed. The only thing that changed was fatty acids overnight. So we then, here you can see fatty acids in the morning, and here you can see after 6 weeks of high-fat diet, the nocturnal fatty acids were increased. So we hypothesized that the signal to the beta cell in the face of insulin resistance was nocturnal fatty acids, and fatty acids at night. And then Dr. Josie Ann Broussard, and I'll always be grateful to her for this, did the same study in humans. And they sampled overnight, and boy, if they didn't find the same change in fatty acids they found in the dogs. 2 different species showed a big increase in fatty acids in the middle of the night. She was studying something else I will discuss, but basically the same time. And in fact, the peak time was the same in dogs as it was in humans. Remarkable. So we then hypothesized, and here's the correlation showing the relationship between nocturnal fatty acids and the insulin response. So we then, another hypothesis, the double sword hypothesis. And the reason this is called the double-edged sword hypothesis is when I thought of it, I happened to be in Spain. I was in a palace. There were a lot of swords around. That's what this is. So the fatty acids go up at night. So what does that mean? Well, they probably contribute to insulin resistance. If your fatty acids are high, you're going to be insulin resistant. But by the same token, hyperinsulinemia. So we think the fatty acids is a double-edged sword. They cause insulin resistance. They may also be a signal to cause beta cell function to compensate for it. We haven't proven this again. We think this is true, but we have to do more work to prove it. So we were able then, from this simple model, to calculate a bunch of different metabolic parameters that you want to know for your own health. Insulin sensitivity, because I didn't go through all the detail. That's what SI is. So if you do this test, you can measure insulin sensitivity, which is the converse of insulin resistance. If you measure one, you measure the other. You can measure the beta cell response, which is AIR, so insulin sensitivity, beta cell response, and the disposition index, and you remember this equation, which is a measure of how well your beta cells compensate for insulin resistance. You also can measure glucose effectiveness, and I'll talk about that in a minute. It's the ability of glucose, independent of dynamic insulin response, to enhance net disappearance. That's partially in the brain, it's partially in other tissues. There are tissues that take up glucose, like the liver, independent of a change in insulin. The liver takes up a lot of glucose. And then the clearance rate, the amount of degradation of insulin. And so, starting a few years ago, we had developed the hyperbolic function, we had defined the disposition index. We and others had done a bunch of studies to confirm it, but there were other factors that come out that we could measure which hadn't yet been implicated. And one was the metabolic clearance, one was the glucose effectiveness, which is the ability of glucose per se, independent of any change in insulin, to suppress glucose production by the liver and enhance glucose uptake by the periphery. And the second was the fractional metabolic clearance of insulin, how much insulin is degraded per unit of time. Because if insulin goes up in the blood, what goes up must come down. Any of you that are studying any hormones, if you're studying parathyroid hormone, if you're studying steroids, whatever it is, there's a clearance rate of these hormones. And that clearance rate can be controlled. So insulin has clearance, and I'm going to talk about insulin clearance in a couple of minutes because it turned out to be very, very interesting. So then we started to think of glucose effectiveness. This is the ability of glucose, as I already told you twice, to stimulate glucose uptake and suppress glucose production. If you inject glucose, and you've seen these kind of curves already, this is just a glucose injection. You see that the pattern of glucose decline, which you turn to basic, has several phases. First, there's a very early phase. This is actually glucose just mixing in the blood, so that happens instantaneously. Then there's a period of time when glucose comes down almost linearly, really exponentially. We call it phase 2. At that time, the insulin effect is not yet expressed. The insulin hasn't gone up much. And remember, there's a delay in insulin action. Insulin has to get into the tissues to do anything. So during that period, insulin is still in the blood. It hasn't gotten into the tissues yet. By about 30 or 40 minutes, insulin gets into the tissues, and you can see very clearly in this data. This data was collected by Marilyn Ader, who is my lifelong colleague at USC and now at Cedars. You can see a very clear acceleration of the drop in glucose due to the insulin going up. So the period before that acceleration is insulin-independent disappearance, and then starting phase 3 is insulin-dependent clearance, and then phase 4 is just a return back to baseline. So here you can see there is a period of glucose effectiveness, which is independent of insulin, and the model gives you a number of things. Now, what is glucose effectiveness? Well, glucose effectiveness is a glucose uptake independent of the insulin response. Remember, it happens before the insulin goes up. Where does it go? A lot of it goes into the liver. People are not aware that after in a glucose tolerance test, if you do an oral glucose tolerance test, if you do an intravenous glucose tolerance test, a lot of the glucose you're shooting in goes into the liver, and that happens independent of the insulin. The glucose itself can enhance its own uptake by the liver through GLUT2 transporters. So what we did was we built yet another model. I apologize for the models, but that's what we did. So here's the biochemical understanding. Here's the glucose going through. This is GLC. That's the glucose going into the blood. And then there's something called glucokinase regulatory protein, which emerges at that time and accelerates glucose's ability to disappear. And then glucose goes on 6-phosphate, goes to glycogen, and a lot of it goes to lactate. Because when you just have a patient lying there or an animal lying there, when you do a glucose injection like this, a lot of the glucose goes into the blood and comes out as lactate. It doesn't all get metabolized because there's no need for oxidative metabolism under resting conditions. So this is what glucose and lactate looks like during those tests. You can see the glucose goes up and it comes down, we talked about before. And then there's a lactate response. This is measuring lactate in blood. Glucose goes up, and then the lactate goes up. Why does it go up? Because the glucose goes into the liver, and then it's converted to lactate. And that takes some time. In fact, the story behind this is that I was at a genetics symposium, and somebody was talking about all these gene markers, many of which mean nothing to me. S2314157939Z, blah, blah, blah. But then somebody said glucokinases. I've heard of that. So I did. This is literally true. I ran out, went to the phone, called Viorica Yunitz, who was one of my, unfortunately she passed away, but she was one of my dearest friends, Vivi Yunitz. And I said, Vivi, do we ever measure lactate? She said, we measure it every time we measure glucose because the machine measures it. He said, well, I predict the lactate is going to go up in 30 minutes and then go down. And that's exactly what happened. So we looked at the lactate kinetics, we built the model, and then we were able to model it in a very simple model, and we'll talk about this, but in the end we were able to measure glucose effectiveness. So now we measured insulin sensitivity or insulin resistance, insulin response, what the beta cell does, and the disposition index, and now glucose effectiveness, which is shown here. Now what this is a population study, a genetic study. There's a family that had a GKRP mutation, and their glucose effectiveness was much higher because they didn't suppress GKRP. It was always in the blood, and glucose disappearance went away really fast. So we confirmed it with this genetic study. And there's another study around it. So glucose effectiveness is the ability of glucose per se to enhance disappearance of glucose independent of a dynamic insulin response. The export of lactate during average GPP appears to be a good surrogate for glucose effectiveness. And a family with GKRP inhibitory variant demonstrated increased glucokinase activity using the model. That's what that meant. Rats injected with FGF1 have increased glucose effectiveness, which is why FGF1 can cure diabetes. I'm sure you know that data. So glucose effectiveness appears to affect hepatic glucose phosphorylation via glucokinase. So now we have another piece of the puzzle. All this took many years. I don't want you to think we did this all in a few months. You know, we did the sensitivity, we did the secretion, and we did the glucose effectiveness. And I'm taking this opportunity of speaking here to try and give you the whole picture. So systems, biology, model. What have we learned as of that time? Excuse me. Talk amongst yourself. You can represent carbohydrate metabolism in a carefully selected simple model. That's the middle. Middle model led to disposition, the most powerful predictor of type 2 diabetes. It led to the definition of what that was. You can measure insulin resistance and beta cell function from simple clinical tests, which I showed you, and the importance of glucose effectiveness in relation to liver glucose utilization and lactate. So these are the things I've talked about so far, but I'm not done. That's okay with me. So one more thing to talk about. Now this is let's talk about what's shown on the right here. When the beta cells secrete insulin, the pancreatic islets secrete insulin, they come out of the pancreas and go right into the liver. They don't make any U-turns, they don't get into the systemic circulation. They go right into the liver. So insulin goes from the pancreas to the liver. When it gets into the liver, half of it is degraded and never appears in the systemic circulation. How weird. We secrete this hormone, it goes into the liver, and half of it disappears. Well, many years ago, there was a meeting, and a famous diabetologist, and oh, I'm sorry, so that's shown here. Insulin coming out of the pancreas, which is this arrow, and half of it goes into the liver, and the other half goes into the systemic circulation. How weird. So I was at this meeting, and this guy, Bob Turner, who did a lot of very important work, said, about half the insulin released by the pancreas is degraded by the liver, never to enter the systemic circulation. Why? What is the reason for this weird kinetics? And he actually asked me this during a meeting. And I said, I don't know, Bob. I'm not God. But in the ensuing years, we've actually studied this question. Unfortunately, Bob Turner died at a fairly young age. He was a brilliant, brilliant investigator. So we did these experiments. We did these experiments. So this is a kind of quirky experiment I'm going to try to explain to you to measure the clearance of insulin by the liver. Now, as Dr. Hooper knows, or anybody that worked in Albuquerque, you can do that using C-peptide. But we did it a different way because this way is an unequivocal way to measure clearance of insulin. This is a dog study, conscious dogs. And we had catheters in the portal vein, which is the vein that goes right into the liver. And then we infused insulin into that vein. So insulin will go into the liver. Some of it's degraded, some of it ends up in the systemic circulation. So half of it or so comes out. We did another experiment. We gave insulin directly into the systemic circulation, but we gave it at half the rate because we knew that when we gave it into the liver, half of it would be degraded. So then only half of it would get into systemic circulation. So we could match the systemic insulin levels by giving it at a full rate into the liver and half rate systemic. I think this is a pretty clever idea. So then we were able to do these experiments. And so we did different doses. And here you can see the doses that we gave systemically, or half the doses that we gave portally, to end up with the same concentrations. And that worked. And these experiments were done in my laboratory, first by Stella Kim, and then by and here you can see what happens. Here's the insulin given it systemically. This is Isaac. Isaac is high. And he gave it either systemically or he gave it directly into the liver. The levels we got were about the same because the systemic infusion was half of the liver infusion. And so if you look at the time course and you plot the infusion rate of insulin versus the insulin level, you get different curves. Different curves because when you give it into the liver, the curve is lower because at the same rate of insulin, you get a lower insulin concentration. So this is a little hard to conceptualize the first time you see it. You can read the paper. But one of the things that was really wonderful was then from this data, I was able to calculate the clearance of insulin because what went into the liver was cleared more than what went into the systemic circulation. And God forbid I should show these equations. But the nice thing about this was that the final equation was very, very simple. You just feel so good when this happens to you. That the clearance of insulin was simply 1 minus the slope of the portal infusion over the slope of the peripheral infusion. You can imagine how I felt when I did this derivation and it came up with this very, very simple equation. Because that allowed us to measure the clearance. So then Stella Kim did these experiments where we did the same, where we simply did clamps and we gave it either peripherally or portally. We looked at the relationship between the insulin level and the insulin concentration, but we also fed the animal fat because we knew if we feed them fat, they'll get more resistant as a function of time. And then we used the method I just discussed to measure the insulin clearance. Well, under controlled conditions, about 60% of the insulin going to the liver was degraded on first pass. 60% was wasted. And then after feeding fat for 6 weeks, that went down to about 56%. So the animal is responding to fat feeding by clearing less of the insulin. Under controlled conditions, it cleared 60% of insulin. After fat feeding, it cleared 56%, but after 12 weeks, it only cleared 43% of the insulin. So feeding fat to the dog reduced its ability to clear insulin. So insulin clearance was not a constant like people thought, but it was a variable depending on environmental conditions, depending on diet. So all of you here who are doing fat feeding or other kinds of dietary studies, you're probably changing insulin clearance. Now whether that has an effect or not is something you can think about yourself. Now the other thing we found, which is quite remarkable in these dogs, not only did you clear a lot of insulin, but the amount cleared by any individual dog was very different from other dogs. These are normal animals. Some of them cleared just, you can see number 8, 22%. Some of them cleared 77%. A huge variance in the clearance of insulin by these animals, even though they were all normal. So all of a sudden, here's something that everyone thought was constant. But if I measured the clearance here, and measured over there, every one of you would have some different amount of clearance. In other words, a different fraction of the insulin that your pancreas is pouring out is being degraded. And some of you are degrading 70%, some of you are degrading 20%. So this is a very different efficiency of insulin secretion. And that's going to turn out to have great significance. This is kind of the last part of the talk. But we're now really interested in insulin clearance because, oh, Jesus, we can change it with diet, and it's variable among different people. So liver insulin clearance, this MCR's clearance, is reduced to the insulin resistant situation, delivering a larger fraction of secreted insulin. This is with the fat free. Wide variation in clearance in normal animals, and we found it in adults too, I can show you. Thus, insulin clearance is a controlled parameter subject to environmental influences. Well, you might say, so what? That doesn't sound very interesting to me. These guys are just at the end of their rope. They've measured everything else. How important is it? Well, then we were able to apply it to humans. And as I say, oy vey, another model. You probably don't know what that means, but if you're interested, so what does this mean? Well, with David Polidori, who's at Janssen, and with Francesca Piccinini, who was in our place, we developed a new model. The details of the model are not important to you, but what the model is able us to do, which isn't true with the C-peptide method, is we could separate out clearance by the liver versus clearance by the rest of the tissues. What I've been showing you up to now is liver clearance. First pass clearance of insulin in the liver. Now we can do it in humans. Well, that was important because now we could ask about clearance of insulin in human subjects. And that turns out to be most of our present work is related to that question. So we looked at the clearance of insulin in human patients. Now, how did we do that? Well, Barbara Gower, of the University of Alabama, had done hundreds of frequently sampled intravenous glucose tolerance tests. Remember, I showed you what FSIGT was a lot earlier. She did a lot of them in people for other reasons. But because she measured insulin and C-peptide at every moment during the test, we could calculate the clearance of insulin in all these people. Again, just like with the dog model, we saw a great variance in the first pass clearance of insulin across the population. So here you can see the variance, where people cleared very little insulin, where people cleared a lot of insulin. This is first pass extraction. And then here is clearance by the rest of the individual. So there's wide variation in clearance among people. But if you look at European Americans versus African Americans, African Americans with the same demographics, except they're African American as the whites, cleared less insulin. And it's very, very different. So here you can see, but they cleared less insulin by the liver, not by the peripheral tissues. Peripheral clearance is not different between African Americans and European Americans. But hepatic first pass clearance, remember, this is first pass clearance of insulin by the liver. And it's extremely low in African Americans compared to European. Well, that's very interesting. We got her data, we were able to do that. So then I walked into Francesca's office one day and said, we ought to see if this is true in children. Because if it's also true in children, we might argue that it's genetic. And so then we found, we were very fortunate that Dr. Fernandez had actually done the same study in 403 children. So we're able to measure first, it's quite an accomplishment, able to measure first pass insulin clearance in children, African American, actually he did Hispanic American and European American. Same result. The clearance of insulin in African Americans was lower than it was in European Americans. Hispanic Americans were between. And then if you look at extrahepatic clearance, it was the same, it wasn't different. So therefore, hepatic extraction is still lower in African Americans compared to whites. This suggests that reduced extraction is genetic or epigenetic. This is the data that was at Fernandez, which we analyzed. So what does this say? There's an ethnic difference in the clearance of insulin. That African Americans have a lower first pass clearance, and as we know, they're hyperinsulinemic. If you measure the insulin in blood of African Americans across the population, it's higher than it is in European Americans. People used to argue that was because they're secreting more insulin. We say no, they're clearing less insulin, particularly on first pass by the blood. Now, what good is this? Well, we said, the question we asked them was, can reduced insulin clearance play a causal role in the pathogenesis of type 2 diabetes? Is the fact that the insulin clearance in the African Americans, can that make them have more diabetes? Because African Americans do have more diabetes than European Americans. So the hypothesis, we've seen that word before, is that reduced degradation of insulin by the liver is the cause of type 2 diabetes. Remember, this is a hypothesis. Don't go up and say, Bergman said it's a hypothesis. We don't know if it's true or not, but we're very interested in exploring it. It may not be true in anybody. It may be true in 5% of the population. But is it possible that reduced clearance, which we have some evidence is genetic, is responsible for the reduced risk of type 2 diabetes in African Americans? So this is the hypothesis. That's the last thing to show you. In people not at risk for diabetes, they secrete insulin by the pancreas. Pancreas makes insulin, and it's degraded about 50% between going to the liver being degraded or entering the systemic circulation. You've heard that enough. Half the insulin is cleared, half of it isn't. This is on the average, because it varies tremendously among different people. When that goes, you guys know this is probably muscle, stimulates glucose uptake, and you end up with normal glucose tolerance. So this is the normal situation. Now what about somebody who's at risk for diabetes? Well, in that case, adipose tissue may have something to do with it, but in any event, the pancreas secretes insulin, goes into the liver, but very little is degraded. A lot of it goes to the systemic circulation because there's less clearance. When it goes there, they end up being hyperinsulinemic because their insulin is too high. Now, what happens when they're hyperinsulinemic? Well, we know that hyperinsulinemia causes insulin resistance. If you make an animal hyperinsulinemic, their insulin sensitivity goes down. This is all hypothetical. So then that could cause prediabetes. So one thing you can tell Dr. Perrault, when you see her, that Bergman said the lower clearance of insulin may have something to do with prediabetes. The clearance is lower, therefore the insulin can be higher. That can cause insulin resistance and possibly diabetes. So in diabetes, the pancreas secretes insulin at the beginning of type 2 diabetes. But again, very little is degraded. Most of it goes into systemic circulation. That goes into muscle, causes insulin resistance, hyperglycemia, and then beta cell failure. Because when you're insulin resistant, it puts a lot of stress on the beta cells. This is our hypothesis of what causes diabetes based on insulin clearance. It was only going through a sequence of these experiments that we ever ended up with this hypothesis. So this is not looking at big data and hoping an hypothesis drops out. This is doing experiments trying to figure out what causes this disease. So the hypothesis, again, is a reduced degradation by the liver is the cause of type 2. It is hypothesis. Maybe in some individuals it needs to be proven. There's a whole variety of questions to answer. So several people asked me today if I'm retiring. It's going to be a little hard because we have to answer these questions. And I won't go through the detail, but I think you can see that once you pose this hypothesis, there are a lot of things you have to test. Will downregulation of insulin clearance lead to diabetes in an experimental model? Because you don't want just associations, you want to show cause and effect. This hypersecretory contributes to hyperinsulinemia in African Americans, et cetera, et cetera. You can read these yourself. Say there's a lot more work to do in our laboratory. We're going to do it. If you want anybody to work on this problem, send them up. So here again is Bob Turner asking this question, and now I can answer him and say, well, liver insulin degradation is a process controlled by environmental and possibly genetic factors. It acts as a controlled gateway for insulin release into the systemic circulation. If degradation is too low, it will be a factor, even a causative factor for type 2 diabetes. And I'm sorry that Dr. Turner isn't around to hear this. So these are the parameters of the phenotype. I've talked about them already. They came from the original FSIGT minimum model. And now we've worked our way all the way to the end to try and figure out the importance of the weight to time. So this is the method that we use with both predictions, experiments, modeling, and a lot of hypotheses. And this is the lab group. Josie, why weren't you here? Oh, it's after you left. Josie should be here, but it's after she left. So these are a lot of our colleagues where we are. You can see this on the American side. In the lower left is Marilyn Ader, and she and I have worked together for decades. She's responsible equally with me for all the studies that we've investigated. We have a lot of people who are clinical and technical and so on and so forth. It's a wonderful group, and a wonderful place at the hospital for the stars. Notes on this.
So if you could further hypothesize, do you talk a little bit about the possibility of exercise or changing liver metabolic status? So I know you did high-fat feeding studies and you showed that liver insulin clearance was changing. Would exercise or fasting do the opposite?
Yeah, I think so.
Insulin low? Yeah.
I absolutely think so. I don't know about fasting. I think exercise would. And you would find increased clearance and less insulin loading in the blood. So it's not just the secretion that's changing, it's also the clearance. And you have to think about both all the time. Yeah.
So isn't there an alternative hypothesis that the liver is protecting the beta cells of the pancreas by clearing less insulin, and there's more insulin available to the periphery to take up the glucose? And the insulin resistance that we talked about with obesity is what's leading to peripheral insulin resistance.
Yeah, I think the trouble is, yeah, I think that's true. I mean, obviously these things are all happening at the same time. So there can be various, quote, reasons, unquote, that this happens. And that can be one of them. So I can't argue with that. I think time will tell whether, when we go and change the clearance directly, maybe we can answer the question. Yeah.
I'm curious if the composition of fat has anything to do with the liver first-pass clearance.
Well, we didn't measure that. I think it's very important. But we just fed a kind of a melange of different kinds of fats. So we haven't looked at that yet, but I assume it would be different.
So just to kind of follow up on that, the composition of fat, African Americans tends to be a little bit higher in saturated fat.
I don't know if that's true in children. Yeah. I mean, I don't know if that's, first place, this is in Alabama, so I'm not sure that the, yeah, why don't you talk? I think that, I don't know for sure. I think it's unlikely that the diet is that different in white and black people in Alabama. In LA, it might be different. Yeah.
So if you think about the percent of liver clearance of insulin as you said, a control parameter that can be higher or lower, what do you feel like are the most important factors that are upstream of that, outside of genetic variation, but in terms of when you have a high-fat diet dogs that are clearing less, how are they getting that signal that they're, how does the liver know to clear less? What signals is it picking up on?
Well, we hypothesize it's also free fatty acids. So, you know, we're big believers in free fatty acids as a signal. So I think that if the free fatty acids go up, that probably affects the liver directly, but we haven't proven that.
That'd be free fatty acids circulating in the circulation.
We should probably wrap up because we're over time.
Let's
but-
wrap up.
Thanks.
All right. So